of Alabama at Birmingham. He is the William D. Jordan Jr. MD Endowed Professor in Vascular Sciences in the University of Alabama at Birmingham, here School of Medicine. He has a long-standing interest in understanding endothelial cell biology, endothelial cell immunology, and angiogenesis in cardiovascular diseases, cancers, cancer stem cells, and Alzheimer's disease. In addition to training biomedical researchers, Dr. Ran has served as a peer reviewer for American Heart Association, NIH, Department of Defense, as well as respected academic journals. His laboratory has been supported by NIH, AHA, American Cancer Society, Breast Cancer Research Foundation of Alabama, UAB O'Neill Comprehensive Cancer Center, and Ann Soap Foundation to conduct biomedical research. The floor is yours, sir.
Okay, thanks, Dr. Mercy, for your nice introduction. I'm honored having this opportunity to introduce the concept of arteriolar angiogenesis and its implications in the development of cancers and ischemic cardiovascular disease. First of all, I'd like to talk a little bit about the history of angiogenesis. In 1787, a British surgeon, Dr. John Hunter, coined the concept of angiogenesis to describe the blood vessel glowing in the reindeer antler. About 200 years later, Dr. Judah Folkman, a surgeon at Harvard Medical School and the Boston Children's Hospital, proposed the concept of tumor angiogenesis when he served the Navy by doing biomedical research. He has found that cancers remain dormant when tumor size is less than 2 millimeters. However, cancers grew crazy once new blood vessels grew into the tumors. He concluded that tumor angiogenesis might be the key to tumor progression. Angiogenesis, in broad sense, includes vasculogenesis, which refers to the formation of new blood vessels from endothelial cell precursors or angioblasts. The usually known angiogenesis by most people, which refers to the formation of a new blood vessels from pre-existing blood vessels or capillaries, usually by splotting morphogenesis, and arteriogenesis. At present, the concept of arteriogenesis that most people know about is the stabilization and maturation of newly glowing blood vessels, collateralization of blood vessels, and the enlarging existing blood vessel lumens as bridges between vascular networks while vascular remodeling. Most cardiologists, including Dr. Wolfgang Schaper, a physician scientist at the Max Planck Institute in Germany and a leader in the field of arteriogenesis, accept this classic concept. The question is there any arteriolar angiogenesis or de novo arteriogenesis under physiological and pathological conditions in adulthood? Before answering this question, I'd like to talk a little bit about the history and the discovery of vascular and cellular growth factor, VEGF, the most important finding in the field of angiogenesis. In 1948, Mattinson hypothesized that a factor X in the retina is responsible for retinal vascular growth. In 1971, Judah Folkman, as I mentioned earlier, the pioneer of in the field of angiogenesis, and the colleagues described a tumor secretory factor that causes angiogenesis as a tumor angiogenesis factor. 12 years later, the lab from Dr. Devolak identified a tumor-secreted vascular permeability factor, VPF, which was later confirmed as the same as the vascular endothelial cell growth factor discovered by Napoleon Falala and his colleagues at Genentech. They cloned and isolated the growth factor and found this growth factor is able to promote the growth of the new blood vessels. When later they showed that if you inhibit the VEGF-induced angiogenesis with monoclonal antibodies, you can inhibit the tumor growth in animals models in vivo. It takes about 30 years for the first anti-angiogenic drug approved by the FDA which can be used for the treatment of metastatic colorectal cancer in combination with chemotherapy. Ever since VEGF was discovered, many types of VEGF and VEGF receptors have been identified subsequently, including VEGF-A, VEGF-B, VEGF-C, VEGF-D, VEGF-E, snake, venom VEGF-F, and placental growth factor. They can initiate signaling by binding with the high affinity to their receptor tolerant kinase, VEGF receptor 1, 2, and 3. The major VEGF signaling receptor in vascular endothelial cells. Whereas VEGF-A plays a key role in the regulation of angiogenesis under pathological and physiological conditions, mainly by interactions with VEGF receptor 2, a principal receptor in the regulation of angiogenesis. As I mentioned earlier, it takes over 30 years for the first anti-angiogenic drug targeting VEGF on market for the treatment of metastatic colon cancers after Judah Folkman proposed the concept of tumor angiogenesis. More and more anti-angiogenic drugs have been approved for cancer treatment by FDA ever since. Unfortunately, current anti-angiogenic therapies, mostly targeting VEGF pathway, do not show significant therapeutic efficacy against cancers as expected. And they also have therapeutic resistance, even likely promoting metastasis due to anti-angiogenic treatment. To overcome the limitations of anti-angiogenic strategies in cancer treatment, it is essential for us to gain a better understanding of angiogenesis and the mechanisms. Since lysophosphatidic acid, LPA, is a lipid signaling mediator that can promote angiogenesis. LPA is also an activator of protein kinase PKD1 that regulates angiogenesis. We performed in vivo major jaw angiogenesis assays by implanting major jaw containing VEGF and LPA into mice subcutaneously. Interestingly and excitingly, as you can see from this result, the major jaw containing VEGF plus LPA appears to form functional blood vessels containing blood cells. And these blood vessels look like small arteries. Based upon our and other studies, I proposed the concept that molecular signaling balance is critical in the regulation of arteriolar angiogenesis. There is an yin and yang balance between pro-angiogenic and anti-angiogenic signaling to control angiogenic phenotype. During this dynamic process. The angiogenic receptors may play a central role in the regulation of angiogenic signaling. Take, for example, VEGF receptor 2 is critical for upregulating VEGF signaling, while CD36 may function as a critical negative regulator in angiogenesis by interacting with an endogenous angiogenesis inhibitor, thrombospondin 1. The principal signaling may dictate the final angiogenic phenotype, in which protein kinase PKD1 may be an important player. PKD1 is a serine-threonine protein kinase, initially known as PKC-mu. PKD1 contains multiple functional domains, including an alanine and proline-rich domain, and the two zinc finger-like structures, an acid region, and the plextrin homology domain. This pH domain is unique in PKD and distinguishes PKD1 from PKC. PKD1 also contains 6 conserved phosphorylation sites. In response to a variety of biological factors, these sites can be phosphorylated to activate PKD1 and regulate such fundamental cellular functions as cell migration, cell proliferation, cell differentiation, and angiogenesis. In addition to regulate angiogenesis, PKD1 signaling also induces stem-like features in breast cancers, pancreatic cancers, and pancreatic neuroendocrine tumors. To define the mechanisms of endothelial cell PKD1 signaling in angiogenesis and breast cancer progression, we have created 2 transgenic mouse lines. In one line, the PKD1 gene is deleted in endothelial cells. In another trapped mouse line, not only PKD1 gene is deleted in the endothelial cells, but there is a genetic tagged GFP in the ribosome of endothelial cells. This allows us to isolate ribosome-bound messenger RNA directly from the endothelium in animals for RNA sequencing and other downstream analysis. We established a marine breast cancer model into the control and the endothelial cell specific PKD1 knockout mice. We show that in the endothelial cell deficient in PKD1, there's a reduction in the tumor growth and tumor metastasis to the lung. And the possibility of the survival of the tumor-bearing mice was significantly higher when PKD1 was deficient in the vascular and cellular tissues. To define the molecular mechanisms, we analyze angiogenic gene expression in the control lungs and the lungs with metastatic lesions. As shown in these results, the lungs with metastasis demonstrated significantly higher expression of pro-arteriogenic genes at transcriptional levels, including PKD1, neuropilin-1, VEGF receptor 2, and such genes in the Notch pathway as like ligand 4 and HA2. In particular, the protein levels of neuropilin-1 and the ligand 4 show high levels in the metastatic lungs. In addition, AL-1A1 that is important for cancer stem-like features and the tumor angiogenesis demonstrate a high expression at both the transcriptional and the translational levels in the lungs with metastatic lesions. When we glow endothelial cells with breast cancer cells, we demonstrated that endothelial cells stimulated the expression of CD24, AL-1A1, and SNL in cancer cells. However, overexpression of PKD1 in the endothelial cells significantly promotes the expression of CD24, CD133, ZIB1, AL-1A1, and Vermentin. We also directly glow the lung endothelial cells with breast cancer cells labeled with GFP. As we can see, some green cancer cells directly interact with the endothelial cells. However, there is little direct interaction of PKD1 deficient endothelial cells with the cancer cells. We then look at the expression of stemness-related and arteriolar angiogenesis-related genes by immunofluorescence microscopy, and we found that the expression of AL-1A1, dialog ligand 4, and NOTCH1 was significantly lowered in the cancer cells cultured with PKD1 deficient endothelial cells. So these studies suggest that cancer cells might positively regulate the development of arteriolar angiogenesis to form the vascular niche in the tumor microenvironment, favorable for the communications of endothelial cells with cancer cells, and the maintenance of the migration of the cancer stem-like cells in tumors. As a matter of fact, in the specimens of breast cancer patient cohort, we have observed small arteries within the tumor microenvironment. Moreover, aggressive breast cancer stem-like cells tend to be accumulated nearby small arteries. Arteriolar angiogenesis within the tumor microenvironment may provide a favorable microenvironment for the expansion of the aggressive cancer stem-like cells, likely leading to metastatic progression. As you can see from this image, we can see that this is smooth muscle stained, actin stained small arteries. Here is the ALDH1A1 positive cancer stem cells like cells. So they tend to be accumulated in the vascular niche for equipment to move to the other places. We also analyzed the single-cell RNA sequencing data generated from breast cancer specimens. Among those single cells that passed the quality control across 3 types of breast cancers, we identified 9 major cell populations by annotating with canonical lineage markers in combination with UMAP visualization. Further analysis reviewed 3 subsets of vascular endothelial cells, including endothelial cell ACKR1, endothelial ACCXCL12, endothelial cell RGS5. We also identified a subset of lymphatic endothelial cell LAV1. And this subset, the endothelial cell ACKR1 was the major subset in all breast cancer subtypes. This subset also accounted for the major population of endothelial cells in the estrogen-positive breast cancer. Although these three groups are subsets of vascular endothelial cells highly express ligand 4, an important marker for arterial differentiation and tip cells. Shadow time trajectory analysis demonstrated their close association, along with CXCL12 positive endothelial ACKR1 subtype showing the highest shadow time score. Both CXCL12 and the ligand 4 are arterial endothelial cell markers critical in angiogenesis. So immunohistochemistry also confirms that endothelial cell ACKR1 in the arteriolar vessels, which also express CXCL12, suggests that some cells from endothelial cells CXCL12 subset have differentiated into the arteriolar endothelial cells in breast cancer. To explore endothelial cell communication with breast cancer cells in tumor progression, we took advantage of cell chatter analysis and found that arteriolar endothelial cell ACKR1 interacted with cancer basal cells. These ACKR1 endothelial cells positive for CXCL12 and the ligand 4 could attract stem cell-like basal cancer cells through the CXCL12, CXCL4 axis, while arterial endothelial cell derived ligand 4 potentially signal those basal cells while Notch 2 and Notch 3 signaling for the phenotypic change in the breast cancer. In summary, our study demonstrates that PKD1 signaling may uniquely regulate arteriolar differentiation of vascular endothelial cells and arteriolar angiogenesis in breast cancers, most likely through ligand 4. Endothelial cell PKD1 signaling may confer stemness features in breast cancer cells, implicating in metastatic progression. Arteriolar endothelial cells may interact with basal-like breast cancer cells through ligand 4 mediated Notch 1-2 signaling in breast cancer progression. Now, I'd like to talk a little bit about arteriolar angiogenesis in ischemic cardiovascular diseases. Ischemic cardiovascular diseases are characterized by reduced blood supply to the heart or the peripheral and the brain tissues due to a blockage in the arteries, leading to tissue and organ damage. Our studies suggest that protein kinase PKD1 signaling may be important in these common diseases through regulation of arteriolar differentiation and arteriolar angiogenesis. Shown here is a piece of data from mouse Handling ischemia model by using laser Doppler imaging and marker CT. And to create the Handling ischemia model, we just simply cut the distal and the proximal femoris and to establish Handling ischemia model. As we can see in this statistical bar graph, compared to the control leg, there's a change of the blood supply between different groups of the animals. As we can see, immediately after the surgery, the blood supply was almost completely disappeared. However, in the wild-type control, the blood supply was gradually recovered and reached around 80 to 90% at day 14. However, in a transgenic animal model that has the impaired arteriogenesis, we can see the blood supply or blood perfusion was recovered only around 60%. Interestingly, when we treated the animals with screened chemical molecules as known as GS4898, which can repair impaired or damaged arteries in a zebrafish model and in the animal models. We can see at day 14 in the groups with in the group of animals treated with GS4898. This is transgenic animal model, as I mentioned, which had impaired arteriogenesis. There's complete recovery of the blood perfusion or blood supply. Although we didn't see significant impact of GS compound on the blood supply in the wild-type control. More interestingly is that if you can see this micro-CT imaging, is interestingly when we treated the transgenic animals with GS4898, which can repair damaged or impaired arteriolar angiogenesis, we can see significantly increase in the numbers of the small arteries with a diameter from 8 to 72 millimeters. More mechanistically, we found the downstream signaling of PKD1 or MAP kinase ERK signaling may be critical for VEGF-induced arteriolar angiogenesis, as shown in endothelial cell with impaired arterial differentiation. As we can see in this Western blot results, the endothelial cell from isolated from the transgenic animals with impaired arteriolar angiogenesis did not respond to the VEGF. However, if we treated this endothelial cell with both GS4898 and VEGF, we can see significantly increased in the phosphorylation of ERK1/2, suggesting ERK1/2 may be a key signaling in the improvement of or control of the arteriolar angiogenesis. Because ERK signaling is downstream of PKD1 signal pathway, so we transduce the constitutively reactive PKD1 labeled with GLP into the endothelial cells. As you can see from these results, when the endothelial cell transduced with the constitutively active PKD1, the shape of the endothelial cells changed significantly. As we can see, this is just one cell. They show an obvious branching morphogenesis. The size was much bigger. In the meantime, we look at the expression of arteriolar genes. We found that the arteriolar genes or arteriologenic genes, including dialog ligand 4 and HEA2, significantly increased, which suggests the correlation of PKD1 signaling with arteriolar angiogenesis. We also performed the three-dimensional matricial angiogenesis assay to determine the function of PKD1 signaling in endothelial cells. When we induced the expression of constitutively active protein kinase PKD1 in endothelial cells by addition of doxycycline, we can see this obvious bronchomorphogenesis. However, if we inhibit PKD1 signaling by PKD1 inhibitor, CID, we can attenuate the bronchomorphogenesis in this in vitro matricial assay. Similarly, when we treated three-dimensional matricial with the LPA, we can see the moderate bronchomorphogenesis in the matricial, which can also be inhibited or attenuated by the inhibition of PKD1 signaling. This is an in vivo matricial angiogenesis assay. We injected the matricial containing VEGF and LPA, or VEGF plus LPA and VEGF and LPA plus LPA antagonist subcutaneously into mice. When we compared these two groups in angiogenesis, we found that there were not significant changes in the vascular density. Interestingly, we observed formation of the functional blood vessels in the matricial containing both VEGF and LPA. These blood vessels could be small arteries, as shown by alpha smooth muscle actin antibody staining. We also examined vascular phenotype in the transgenic mice using the immunohistochemistry charcoal staining. It seems that the integrity of the vascular endothelium in the heart and the lung is moderately impaired in the transgenic animals, where PKD1 is deficient in the vascular endothelium. As you can see, the vascular endothelium appears to be irregular and have defective endothelial cell coverage when compared with the control. We have also established the Huntington ischemia model in transgenic mice with endothelial cell specific PKD1 deficiency. And we found that PKD1 gene deficiency in the vascular endothelium when the cilia impaired the blood perfusion capacity. As here, maybe this figure is more understandable as we shown before. As you can see, immediately after surgery, the blood supply was totally disappeared, almost totally disappeared, as shown by this blue color. However, in the control animals, we can see the blood flow was gradually recovered, as shown by the red color. Especially at 14, you can see significant recovery in the blood supply. And this is the day 21. However, when you see in the PKD1 knockout mice, this endothelial specific PKD1 knockout mice, you can see immediately after surgery, the similar. However, this blood flow was gradually recovered, but this recovery was impaired. Even at the day 21, we didn't see significant recovery in the blood perfusion, as shown by this blue, maybe a little bit yellow color. And specific PKD1 knockout 12 months showed a similar trend. Here is the bar graph. You can see, even at day 21, only around 40% or 10% of recovery of blood supply in the animals with PKD1 deficient in the vascular endothelial cells. And also, you can see here, in the endothelial PKD1 knockout mice, there's necrosis as shown. The nail is black. That means there's necrosis in the handling vessels. Intracranially, charcoal staining in gastrocnemius muscle in the injured leg demonstrated extensive collagen staining at day 7 in the endothelial cell specific PKD1 deficient mice, suggesting the development of fibrosis, vascular leakage, and increased inflammation in the ischemic limb due to deficiency in endothelial cell PKD1 signaling. We also performed immunofluorescence staining of leg muscles with CD36 stained green and alpha smooth muscle actin stained red. It seems that the coverage of smooth muscle cells was impaired in the endothelial cell PKD1 deficient leg blood vessels. There's also irregular capillary formation in the endothelial cell specific PKD1 deficient mice. Here you can see the coverage of smooth muscle cells was impaired. In summary, our study demonstrates that angiogenesis occurs under pathological conditions in the adulthood. Protein kinase PKD1 and MAP kinase ERK1/2 signaling may play a key role in the development of arteriolar angiogenesis. In addition, PKD1 signaling mediated downregulation of CD36 may provide the molecular hub to regulate arteriolar angiogenesis by pro-arteriogenic reprogramming in vascular endothelial cells. So I do not show much data related to molecular mechanisms in the regulation for arteriolar angiogenesis. Shown here is the working model based upon our and other studies, in which PKD1 may be a central player. Moreover, the signaling regulation of arteriolar angiogenesis may be implicated in the development of an anti-angiogenic immunotherapy. I'd like to thank my lab members for their contribution, my collaborators, Dorothy and Darren, for performing Hanlimese chemical experiments, Dr. Peterson, Dr. Zhang, and Dr. Chen for their reagents, data analysis, and helpful input. I'm also grateful for the mentorship from Dr. Kozlova, Dr. Laura, Dr. Sams, and Dr. Silverstein. Finally, I'd like to appreciate the support from different resources. This generous financial support makes my research possible. Thank you very much for your attention.
Thank you, Professor Benran. Truly appreciate it. One question came in. How about the different endothelial cell subtypes? Difference in the cancer. Which one is more important?
Yeah, I think endothelial cells are heterogeneous. In the tumors, also we have different types of endothelial cells. Integrally, in our analysis of the breast cancer endothelial cells for the single cell RNA-seq data, we found that there's most endothelial cells are tip cell-like cells, and also arteriolar endothelial cells accounts for the major population in this breast cancer. We also, as we can see from here, we also found that in the ER-positive breast cancer samples, human samples, we have more endothelial cells. And also this endothelial cell, we call the endothelial cell ACKR1. This endothelial cell also express CXCL12. Maybe they have more stronger immune property or traits. So we are wondering why it happened this way. Maybe other endothelial cells, they might tip cells, they might venous endothelial cells. We don't know, but we know that the endothelials are heterogeneous. Maybe depends on the what markers we are going we select, especially in the normal endothelials in the past. Endothelials we just classified venous endothelial cells, arterial endothelial cell, and the capillary endothelial cells. But now we have many markers. We use the markers since we found this is endothelial ACKR1 cells is exclusively in the breast cancer. I checked like brain tissues or other tissue. I didn't find this subtype.
Thank you. Thank you. For the PKD1 knockout assays, why did you choose the day 3, 7, 14, and 21?
Yeah, this is kind of in cardiology, we select every 3 days. Usually, it's supposed that 14 days, most the blood supply will recover in the wild type mice around maybe 80 to 100. Depends on the surgery. The surgery kind of there's a different because you cut the you cut the femorals. Maybe there's some there's some difference between different labs, but generally 14 days you can see the phenotype. Wild type can be recovered. So 7 days because 7 days in the middle of that we can see the difference between endothelial what changed in the blood vessel is kind of in the middle. Most labs select 14 days. Later we even because the damage in PKD1 knockout mice is very dramatic, we extend to 21 days.
Thank you. How about the FOXC2 related to the PKD1 in the arterial angiogenesis in cancer?
I beg your pardon? Which one?
FOXC2. Related to
FOXC. Okay. Yeah, FOXC actually, Tumming lab, I think they report FOXC is important in the regulation of the arterial differentiation. So, but from I didn't look at FOXC2 in tumor microenvironment. It might also involved in FOXC2. But in our study, interestingly, we found FOXO1. FOXO1 actually not only regulates the stem cell property, we found FOXO1 regulates the expression of arterial like gene expression. So maybe involved in the stem cell like endothelial progenitor differentiation into the arterial endothelial cells. I don't know. We have some data I didn't show here. We this kind of transduction, the forced expression of FOXO1. We don't know, in vivo. But FOXO1, I think that's reported more than 10 years ago. They showed that FOXO1 might be involved in the tumor angiogenesis. I'm not sure about the endothelials. We don't know.
Thank you. Can we control the direction and branching of angiogenesis in vitro or in vivo by controlling the level of each marker?
It's possible. I don't know. It's very interesting when we transduce the PKD1 into endothelial cell, we see that that's huge cells. This is very, I don't know what happened, but when we transduce the PKD1 into the endothelial cell, we saw a lot of branch morphogenesis. So, yeah, where is this? Okay, oh here. You see, this is one cell actually. It's very huge. This 100 is very big and big endothelial cells. They have a lot of branch angiogenesis. But interestingly, we look at the ligand 4 increase because in the field of angiogenesis, most people think that ligand 4 is a T cell marker. Then they said it's a T cell marker. Then when you form the T cell, this ligand 4 is disappeared. But for us, we found that actually ligand 4, ligand 4 actually not only the T cell marker is important for the arterial differentiation. So then I searched the literature. Actually, in gene and development, it has reported that ligand 4 is an important molecule to determine the arterial differentiation, especially during development. So yeah, I'm not sure about that.
Thank you.
Yeah.
Thank you. Thank you for the interesting talk. I previously attempted to knock out the PKD1 gene in the context of autosomal dominant polycystic kidney disease, PKD. I'm curious how you design your experiments involving PKD1. Specifically, how did you come to associate this gene with breast cancer? Was there a particular pathway or mechanistic insight that led you to this connection?
So PKD, that's different. Polycystic kidney disease, that's different. This PKD is a kinase. It's different concept. That PKD is cysteine kidney PK. Kidney, that's different PKD, although the abbreviation is the same. What we talk about is this PKD is a kinase, is a serine threonine kinase, originally known as PKC-mu. That's a different concept. But what we think that protein kinase PKD1 can activate the MAP kinase signaling pathway and induce the arterial differentiation of endothelial cells in tumor microenvironment through ligand 4. This ligand 4 can interact with the cancer stem cell-like cells, like basal cell-like cells. They induce the transformation of the basal cell-like cells into the real cancer stem cell-like cells, then leading to the tumor initiation and metastasis. In our model system, we saw that it mostly affects the lung metastasis. We are waiting for the NIH funding for the study. Actually, yeah, hopefully we can get this funding as soon as possible.
Thank you. Well, that's all the questions I have. Thank you for your time, Professor. Appreciate it.
Okay, thank you.